Good morning, everybody. Clearly, I cannot rely on the microphone to get order in the room. My name is Kostas Tsatsoulis. I'm the Vice Chancellor of Research and Dean of Graduate Studies here at S&T, and I'd like to welcome you all to the 2019 Research Summit. I'm excited to be hosting it for the first time, and I'm excited to see the response that we got from our 4 system universities who wanted to participate in the summit called Engineering and Health. I think it addresses a lot of the research that's going on. We actually had to turn people down. So I hope that there will be equally exciting participation in the summits that we're hosting in the future across our system universities. First of all, I'd like to welcome members of the Board of Curators who are here to observe, members of the administration from the 4 sister universities, and of course the faculty who are giving up a lot of their valuable time to come here and listen to our presenters. And of course the presenters themselves. I'd like to thank them for some of them making the very, very long trip from Kansas City, 4 hours. I just drove today from St. Louis, and so I'm glad that we can all come here and join in Rolla. The goal of this is not to talk about what you did over the summer. The goal of this, and that's why we're holding it to 5 minutes, is to be able to hear what everybody else is doing, what everybody else is hoping to do, and build collaborations. And I hope that there will be a lot of conversations after the talks over lunch, and I'm looking forward to seeing a lot of proposals with subcontracts across all our 4 system campuses. Well, that's my job, right? I also know that if I am in front of the microphone, I need to be disciplined enough to get off, so I will do that now. We'll start with the presentations, and I will be keeping time. We'll try to keep it at 5 minutes. When there are 2 minutes left, I'll raise my arm and go like this. And if you go way beyond 5 minutes, I'll get up, and this has wheels, so I just move it. And I'm serious though, be careful, don't lean on it because I did that. So without any further ado, let me introduce Badri Adhikari, Deep Learning Science New Hope on Solving the Past Century-Old Problem of Protein Folding from I'm sorry. One moment, sir. We'll be right back.
Testing 1 2. Testing 1 2 1 2.
Check 1 2 1 2. We may have to go without.
So you said go without an audio.
Yeah. Keep this one here. So all right, folks. The speaker here seems to have a bit of a loop that belongs to anchors. I'm sure that I talked to somebody about that. See if we can get them swapped out. There's a secondary mic on top of it. You see that's what's feeding the live stream, the recording, so everybody online can hear us. So we're going to keep the mic there. Please continue to use it if it's on. And I'm going to kill this morning buzz. Thank you.
All right, so good morning, everyone. So I'm going to talk about how deep learning has shined new hopes on solving half a century-old problem of protein folding. At the heart of the protein folding problem lies a problem known as contact prediction, or protein contact prediction. So if we can precisely predict contacts, this will lead to accurate prediction of structures and function in proteins, and also this will eventually lead to curing diseases through drug design, better understanding of how life works, and also will lead to improvements in machine learning and deep learning because the contact prediction problem is a difficult one. So the contact prediction problem is given a sequence of protein, amino acid sequence. We would like to predict which amino acids interact with which. We would like to come up with a map like this that shows the pairwise interaction or not potential between every possible pair of amino acids. So the challenge in solving this problem of contact prediction is known as transitive noise. That's the biggest roadblock in solving this problem. So if two amino acids, say A and B, are actually in contact, and A and D are in contact, and D and C are in contact, then when you do your calculations, when you look at the data, what you will see is B and C are also correlated, or they are in contact. So the challenge is removing this noise, known as transitive noise, in your data. So can we write algorithms to predict contacts or remove this transitive noise? And it turns out we can. So there are many algorithms that people have written, which are standalone tools that we can download and run it to predict contacts. So now the next question is, can we apply machine learning or deep learning to do the same thing and do it better, right? And the answer is yes. So in this plot, what you see is in red are the algorithms that are not based on deep learning, and the green ones are the algorithms that use deep learning methods to remove transitive noise and predict contacts. The DeepCon method is the one that we recently developed. It's an archive and it's under review. But overall, we see that all deep learning based methods are better than the algorithms that are based without deep learning. So in the most recent CASP competition, which is like the top competition for evaluating the best methods for contact prediction and structure prediction, the top methods are Google's DeepMind, University College London's Deep Metapsychove, and University of Michigan's Residual, all had one thing in common, and that was they used something known as residual networks, a variant of deep learning methods to solve the problem. So the immediate question is, what variations of this architecture or of this deep learning architecture are best fit for solving the problem? To answer this question, we need to try almost all possible architectures, which means a lot of resources, which means a lot of GPUs, basically. The input data is ranges anywhere from 2 gigabytes to 200 gigabytes. That means if we want to do the training in a decent amount of time, we need to read 200 gigabytes of data within 20 minutes. And that is not possible in the Lewis cluster that we currently have. We need a special hard drive that we can plug into our machines and can be used to read data faster. I applied to Google for resources to see if the resources are helpful. They immediately gave me with $5,000 as a startup fund. We finished them within a week. I asked for more and they gave 5,000 more, which we again finished it in a very short period of time. So that wasn't very helpful. I applied to NVIDIA for GPU resources and they gave me a Quadro P6000 GPU, the best GPU, one of the top GPUs. And this has been very, very helpful. We do most of our research in this GPU. And we tested various architectures and we finally came up with a deep learning architecture that's best fit for this problem. So we tested fully connected, fully convolutional networks. That's the FCN. We tested all other variants that other people had been using to solve the same problem, residual architectures. Finally, we found out that combination of residual architectures with dropout and isolation are best for solving the problem. So, but is there any room for improvement? Yes. In the most recent CASP competition, this is what one of the senior scientists had to say. It was good to see Google DeepMind win this time. I was sick of seeing Rosetta win since almost 20 decades, right? Overall, we are very far from still doing something known as end-to-end deep learning, where you can feed the sequence and deep learning will figure out the answer for you. So in conclusion, overall in the field over the last 20 years, what we see is groups who are good at exploring new flavors. Those who are like, trying new deep learning methods and all of that did very well. So in other problems, it may be a good idea to try new deep learning methods and learn them even when you don't see a direct fit to your problem. Balanced efforts of machine learning experts and domain experts brought a lot of success in this field, right? So that may be something to look at. And when end-to-end deep learning is not possible, that is when machine learning cannot take care of your problem right away, you have to do feature engineering. That means correct feature engineering is extremely important to solve your problem. And say, for example, if you have images as input, then you don't need to do any feature engineering at all. You can directly feed images to machine learning pipeline and it should take care of that. With that, I'd like to thank everybody who contributed in my research, including my graduate students and computing resources provided by Google, NVIDIA, and UM system resources, even though they are frequently down and all of that. But yeah, thank you all.
Thank you. Next talk is by Giovanna Guidoboni. Quantitative cardiovascular and lymphatic research for diagnosis and treatment of disease. Dr. Guidoboni is also from University of Missouri.
Thank you. So thank you for giving me the opportunity to present this research. So the cardiovascular and lymphatic systems, so they work together to move fluid and oxygen and proteins all around our body. So it is not surprising that if they do not function correctly, many pathological conditions actually pop up. Well, except for, you know, being at the top of the list of the worst diseases that affect basically the world and Missouri, what we are particularly interested in is trying to help with early diagnosis and monitoring of the progression of these diseases. And what are the main challenges we are addressing? So first of all, even the same cardiovascular disease manifests differently in different individuals, in particular, for example, among men and women. The lymphatic function that contributes to the motion of fluid and also the function of the cardiovascular system has been widely understudied. And so a lot is not known about how it functions even in healthy situations. And moreover, what we are particularly working to kind of change is the fact that research has been kind of conducted in a reductionist way, and that it makes it really difficult to interpret the data. So my next slide is to elaborate this concept. So our life is truly happening on multiple scales, both in time and in space. So reductionist, I mean that maybe first, you know, we focused on the behavior of the body to see if something is wrong. And then depending on the scientist, the focus might have been more on the organ or on the cell or down to the protein exchanges on the cellular membrane and down to the genes. But actually, our system works all together, and the challenge is to put these scales together, because indeed, for example, if we run, then our heart knows that it has to pump more, but then the signal, what is actually is making it pump more is what happens at the level of the cells, and we see the effect at the macro scale. So what are the challenges? So we know that we need to do this, but this reductionist approach has taken us to deal with these problems in different compartments. So the classical ways to study problems related to life and health has been based on clinical studies on people. And of course, you have real data on humans, but you cannot do anything too invasive, of course. On animals, you can do more, but still it is a living system. So every time you try to measure something, you affect something else. So it is difficult to isolate the factors. And when you go down to the cells in vitro, of course, they are not in vivo anymore. And so the question is, you know, are we maintaining the features that are there actually in the living body? So to help address these challenges, in Columbia, we are trying to build a multidisciplinary team of scientists where we try to bring in the physiology and the biological aspects on how the system functions at different levels, together with the engineering and mathematical aspects of bringing the physics of how blood moves inside the model that then can help the data-driven models in computer science to take into account of the variability that you have among the data in individuals. So it is a combination of data-driven and physically based modeling that can kind of connect the dots among the different disciplines. So very quickly, just a few examples. One is on the eye. Many things show up in the eye because it is transparent, but the difficulty in unlocking it is that the measurements are done at different sites. And so we are working on using math and physics and computer science to bridge the gaps. So for example, the doctors can measure something at the level of the central retinal artery and vein, but what they're really interested in is what happens here at the back of the eye. And so by combining simulations and data collected on patients, we can give them an estimate of what cannot be accessed directly based on what they can see. And the last example, of which Marge will talk more about at the end of the day, is related to monitoring the cardiovascular progression of diseases. And so maybe we are all familiar with the ECG that captures the cardiovascular the electrical activity of the heart. But Marge and the group at EECS have developed many sensors that can passively capture the motion of the body based on the blood moving. And the fact is that the signal is not standardized, and we are using math and the modeling of physiology to provide doctors with a waveform that they can interpret at a similar way as the ECG. So this work is indeed collaborative because it brings people from different domains. And here you see in colors coded basically the different departments. And what I would like to advertise is that we are conducting monthly meetings for which really I would be very happy if there are participants from other campuses, because they are available in Zoom and also recorded in Canvas. The idea is that we kind of help each other entering each other's domain. And so the lectures are really for us to understand our language and communicate. So thank you for your attention.
Thank you. I will ask if the presenters can remember to use the microphone and this thing to move the slides so that the people who are watching us can actually see us. The next presenter is Dr. Zhu Li from UMKC. The talk is what NSF Center for Big Data and Learning can do for you.
Okay, all right, thank you. Good morning, everyone. It's my pleasure and honor to present this recently funded NSF Center for you and what are the opportunities it brought. So this NSF Center is part of this NSF IUCRC scheme. It's Industry University Collaborative Research Center kind of scheme, very successful for the last 40 years. So we are kind of it's the first kind of NSF National Center on Big Data and Deep Learning. Because originally we want to name to Big Data and Deep Learning, and the program that director for that name is just 2 now. So we shortened them to Big Learning. So we started with 4 universities. So Florida is more focused on the cloud computing infrastructure for deep learning, and the CMU is going to be very good at this. Unfortunately, they dropped out because the IUCRC scheme is not cost efficient for them, because their membership cannot support half student at the CMU. And then for University of Oregon has expertise in the health informatics. At UMKC, we brought in expertise for deep learning for embedded system, basically imaging, compression, as well as computer vision tasks. So starting this year, we'll bring the Northwestern University into the center. So headed up by Professor Katsagas. He's heading up the. Oh, sorry. He's heading up. Okay. So he's heading up a new sort of group for AI in medicine at Northwestern. Basically, very much like what we are trying to do here, combining the engineering school and the medical school expertise in driving new frontiers in the research. So the center is funded for $3 million from NSF, but collectively we have to raise $3 million from industry for the next 5 years for the center to be viable. So for the first year, it has been very successful. So these are the center kind of leadership. So on UMKC side is me and Yugi. I think Yugi was at this summit last year. He probably gave some instruction as well. And starting next year, we have actually studying this year. So Northwestern will bring quite a bit of a strong group, exclusively focusing on the medical side, and also Professor Yungman Noor from KAIST, also a very kind of powerful institution in Korea. They will be also joining from working on the explainable kind of deep learning. So deep learning, a lot of people were very negative against that, even though it works, does not have a framework to explain why it works. So Professor Yungman Noor's group is trying to give it a rigorous math framework to predict the performance, this and that. So overall the center is very healthy at this time. So we were supported by around 30 kind of industry members. So this IUCRC runs as if it's a mini NSF within the university system. So you can sort of solicit kind of proposals from faculties. But the other side of the coin is you have to raise kind of resource from the industry members. The idea is that hopefully, as you produce more and more results and get the industry excited, it will become self-sustaining. So, okay. So at this time, we are focusing on a very diverse kind of range of topics related to big data and deep learning. So starting from the sensor side and then from the beginning side, ranging from auto driving, media, medical system, this and that. So UMKC side, we have quite a bit of kind of faculty involved. So, I mean, we very much want to involve kind of colleagues from other campuses to join this center and to work together. So at this time, externally, we have, I mean, on my side, I work on quite a bit of 3D sensing, imaging, and the computer vision kind of problems. So we have a collaboration with the Air Force Academy, and that gave us quite a bit of kind of resource and insight into the DoD side kind of problems. And so the first year has been very successful for UMKC. So we raised around 200K from industry for the first year. So that was matched by 150K from the NSF side. So we had a very interesting project on 3D sensing, mobile edge 3D sensing, and low latency point cloud service to the users, to the auto driving and the 3D map application. And also we also had a very impressive work on use deep learning to enhance very dark images. So basically, you have pixel in, pixel out, right? So how do you sort of play this magic? So for the sort of surveillance, it's dark image, low resolution image, kind of a hazy image, right? For medical side, is mostly kind of enhanced resolution. It's the same type of technical setup, but different applications. We also have a knowledge graph, and it was done by Praveen. And also, we also have a very good work on the immersive media side. So basically give you a 3D experience. So light field, 360 camera, 360 camera video compression, as well as deep learning in improving the existing compression pipeline. So this is a 5-year kind of roadmap. I mean, it's too hard to read, but in general, we are building all those big hammers with deep learning and kind of big data kind of tools. We want to kind of deliver value and applications to a variety of applications. So what this is useful is now the center is up and running. We have quite a bit of momentum. So we want to involve more faculties from the UM system. So basically, I mean, we can bring your proposal to the meeting towards the end of May. We also kind of encourage you to help us link up with industry, find out what they need, and also bring kind of membership. So I mean, quite a bit of the details are in that link. I think that's accessible. Okay, so that's very much it. So it's a kind of a building kind of vehicle for us to expand our research outreach and get more resources. Yeah, thank you.
Thank you. Our next presenter is Dr. Laura Shop from Mizzou. I will let you read the title of your talk.
I made it simple. Actually, I'm having a little hard time.
Okay, I'm sorry.
The screen on the right is a little out of focus. On our right.
Okay.
I'm going to move over so I can.
Okay. Hello, good morning. I want to thank our partners here at S&T for being such great hosts to this summit. My talk today, I have a simpler title than what you have. I call it You've Seen One, You've Seen One. I just went backward. Let's see. All right. I call it You've Seen One, You've Seen One, targeting treatment for diverse patient populations. And I hope to get you excited and concerned about the same problems that we're concerned about. So what is the problem? We know that NIH is going to invest about $39.2 billion each year on health research. That goes up and down, but it's a lot of money. And unfortunately, many of the clinical trials that NIH supports, they fail to show effects because they're doing treatment group and control group, and there's too much heterogeneity within the treatment group. They contain very mixed samples, and those individual differences within the treatment group can completely wash out the between group differences. treatment effects. And it's a huge problem in all health research, but it's also a problem in education. It's also a problem in policy. It's a fundamental problem in the philosophy of science. So we need systematic tools to identify those homogeneous populations within broad heterogeneous groups. All sectors of research, all of us need this, and it is the very foundation of a successful approach to precision medicine. So let's talk about what happens when we throw spaghetti at the fridge and hope that the patient gets better. One-size-fits-all treatment does not work for our patients. The U.S. spent $3.5 trillion. It is about 18% of our GDP, and we spend more than anyone else in the world on healthcare. Our Missouri healthcare spending has risen by almost 6% every year since 1991. Unfortunately, among those developed nations, we have the lowest life expectancy. We have the highest infant mortality. We're not getting bang for our buck. Why? And so what we hope to do is to put in some tools in place to help us customize our care better. It's a big problem with lots of facets, but we'll talk about one element of the solution. We do know that when we throw spaghetti at the fridge and hope the patient gets better, patients and families bear the cost. There's enormous financial cost, there's side effects and toxicity, and eventually patients get tired. They quit treatment. There's treatment fatigue and people bow out. With respect to targeting treatment, I want to give you an example or two. Let's talk about Alzheimer's disease. About $1.4 billion in NIH spending in fiscal year 19 just on Alzheimer's disease research. In 2015, the mortality for Alzheimer's disease was about 135,000 people. It's going to get bigger as we get older. I'm not getting any younger, and I don't know about you, but we do know that there are distinct subtypes of Alzheimer's disease. If we're just looking at it from the point of view of imaging, we know there's at least 4 subtypes, right? There's a typical Alzheimer's disease pattern, and we can see that in neuroimaging. There's a limbic predominant variant that hits the limbic system, very primitive functions in the brain. There's a hippocampal sparing one. Hippocampus is what helps us remember things. And there's a variant that seems to spare the hippocampus, but affects other parts of the brain. And then there's another kind with no atrophy that's visible on neuroimaging, and yet it continues to produce impairment in functional living. So we know that from the neuroimaging perspective, there's at least 4 subtypes. If you look at it histologically, there's more and different ways of grouping and scheming these folks. We do know that if we can identify subgroups for targeted studies, we will understand how best to approach each person with their different variants of Alzheimer's disease. Let's talk about depression. $437 million in NIH funding for fiscal year 19 going to depression. Mortality is over 15,000 people per year with death related to depression. We spend $210 billion in health and productivity costs. It's a very serious problem, but we also know that there are subtypes within that. There's a cranky, depressed person, very irritable. There's the withdrawn, vegetative person who just doesn't have a lot of motor movement, has a great deal of fatigue. There's depression secondary to heart bypass. We know that's very predictable, and they all look different, and we treat them all the same. It's a problem. And we also know that about 20 to 40% of people with depression get better on their own. So are we over-treating that group? How do we identify the right people for the right treatment? And where do we go from here? One superpower I have, and it's only one, is to identify really intelligent people who can solve clinical problems that I have. And I'm fortunate to partner with Shiren Xu. He just led an R01 proposal that went in the fall, where we hope to identify, to use machine learning techniques and algorithms to identify homogeneous subpopulations within heterogeneous larger populations. So we'll look at things like demographics, environmental factors, symptom characteristics, genotype, onset pattern, course. All these things are wildly different even within people with classic diagnosable conditions. And then we'll apply these machine learning tools and algorithms to new populations. You can think of heart disease, substance abuse, critical area for us. Parkinson's disease, arthritis, hypertension, you name it. There's application of these kinds of tools. So with that, I want to introduce my collaborator and the smart guy that I got to work with on this project, Shiren Xu.
Okay, I'm going to talk about part 2 here. Okay, so I'm going to talk about more like, geek language about what is explainable AI, actionable AI. I call that X square AI for cohort discovery. I'm from Columbia campus. And then if you look at this chart here, I think Laura did a wonderful job to talk about how we treat our patients, what is the effectiveness of the drug here, right? So if you can look at one of them here, the best one is Humira. It's 1 out of 4 really effective to receive the treatment. It's a drug here. If you look at number 2, Nexium, that's 1 out of 25. It's better not to give this patient drug, right? Because you think about this way here, the side effect is going to really overweight the effectiveness of the treatment here. So our hope is to find subpopulation, and we hope to turn all those red patients into blue patients. And that's the hope for that here. So how we do that here? Let's look at the data-driven idea here. It's can we build a robot just going to mimic physicians, and then to look at panel of the patients instead of your panel of patients. That would be the entire collection from worldwide here, and try to slice and dice. Just like in clinical trials, we try to do inclusion and exclusion criteria, and getting into the sub-cohort that really can show the potential effectiveness of the treatment for that. So how we develop this one here. So let's look at this space here, just like your population space here. And then that could be age, could be all different comorbidity idea here. And then if I can identify a pair of the cohorts, that is treatment as well as the control group here. And then if you look at the small one here, AB together could be biomarkers for a treatment group here. And then you see from this cohort, but you don't see from the control cohort, you don't see from the rest of the patients for that, right? So the hope is are we able to find those cohorts? And are we able to find those contrasts between cohorts so we can provide more actionable plan for those patients here? You look at this one here, big cohorts here, and you can have small cohorts here. Big cohorts here, we are talking about population health. Small cohort here, we're talking about precision medicine. Could be tailored to rare diseases. So that's the big concept, the basic concept of this one here. So we developed so-called guided cascading shotgun approach for expansion algorithm that is applying machine learning from traditional floating selection idea all the way to expand deep mining. It's not deep learning, deep mining because of the computational power for this one here. So you look at this way here, we are trying to look at the cohort. The computer is going to look at the cohort here, and then just trying to do inclusion and exclusion criteria. And then if you look at this way, just like shotgun here, right? You got bullet and come in as pellets. On pellets, become a bullet and then become pellets. And then how we cut down the search space. And then how we look for not a single solution, but look for a bunch of solutions. And then we prioritize them based on how actionable they are here. So we apply this one to autism research. Autism is a spectrum. It's not autism or non-autism. Shouldn't be that easy, right? That'd be autism with some subgroup related to social impairment or repetitive behavior or communication difficulty. Those are 3 major phenotypes here. You can also look at subphenotypes here, right? So are we able to develop some treatment to help those subgroups of the patients? So by using this type of the exploratory mining idea, we are looking for, are we able to find new genes that have been discovered? And those genes dedicated to very small subgroup, not really very small, very dedicated subgroup, we can provide treatment for that. So we identified 193 novel genes in this study here. And why they are important here is like we fit them into so-called pathway. Pathway, you look at gene, disease. So are we able to find some treatment already have been proven efficient in different neural disorder diseases for that here, right? So we can talk about drug repositioning, drug like redesign for this one here. So like these days we can see FDA approved many like new drugs coming up for autism. They are modified from like Alzheimer's diseases idea. So are we able to find those subcohort and then finding those biomarkers that can really help like those subcohort of patients? So my collaborators like for this type of the cohort discovery and also like study from different areas here. Lymphedema from Dr. Jen Armour from School of Nursing. For leukemia research from Dr. Richard Hammer from Pathology Department. And colorectal cancer, like immunotherapy study from Dr. Jonathan Mitchum from Surgery Department, and Judy Miles, my collaborator for autism, and also my collaborator from with Laura for the research. And then the innovation in machine learning and knowledge discovery and AI algorithms really important, and we have to make sure they are explainable. And this work is able to create hypotheses for like data-driven hypothesis for further clinical trials. And with biggest population study, and we can also go for precision medicine small population study. So I hope we can collaborate with our UMKC School of Medicine and also pharmacy school to work on this type of the subcohort discovery. Thank you.
Right. Next, I'd like to introduce Dr. Umit Tokac from UMSL. The title of his presentation is on his screen.
So it's such a long title, but believe me, I will try my best to explain everything in 5 minutes. So partial autonomic decision process to implement response to intervention framework for veterans with post-traumatic stress disorder and personality disorders. So PTSD so someone has a PTSD, usually they have the personality disorder too. So over 30% of the patients do not respond to PTSD treatment because of they also have the personality disorder. And therefore, it's really hard to know what kind of treatment will be effective on those patients. Some of the clinicians prefer to apply the PTSD treatment because they prefer to apply it because of the low cost. However, integrated PTSD, PD treatments more intense and twice as effective in the reducing PTSD symptoms than treating PTSD and PD alone. But of course, this is an intense treatment methods is a little bit cost. PTSD also one of the biggest veteran health problems is like almost over 15% of the veterans who serve in Afghanistan and Iraq has the PTSD, have the PTSD. Currently, shared decision-making process applied in military to help those patients. How it works, basically clinician and patient get together and discuss what kind of treatment will be better for that patient. Basically, it's patient preference and needs they consider. However, receiving timely, appropriate treatment could minimize and eliminate the PTSD symptom in a short amount of time with a low cost, and enable veterans to return to civilian life more quickly than the practice shared decision-making process. So what's the response to intervention methods, RTI? RTI has been applied in education such a long time and stays really successful methods. Basically, it identifies students with the reading difficulties or math difficulties by considering lack of progress, determine appropriate instruction and intervention. Basically, teachers consider apply this method to figure out all those students with the reading difficulties and change instruction, apply more intense instruction for those students. In this project, I am planning to apply RTR to identify veterans. They have a lack of progress or slow rate of growth, determine appropriate treatment. So basically, the clinician will apply their overall assessment based on the assessment result. If the treatment is not working, the clinician will assign the patient to tier 2, which is the PTSD PD treatments, more intense treatment. And then whatever the patient has a success, growth, or progress in the general treatment, they will keep getting the same kind of treatment in tier 1. After observing Marko decision process, the process has been applied in economics and robotics and military. The POMD includes probabilistic sequential decision theoretical approach and involves individualized treatment activities. Specific component of POMD includes periodic assessments. So basically, you can use those assessments to monitor the effect of the treatment on each patient. The veteran's current state and applied treatment jointly determine a probability distribution over the veteran's possible next state. So this is the model I want to apply in real life. Basically, this is the first, at the beginning of the treatment, clinician will apply general treatment, which is the PTSD treatment, for a while, like for 2 weeks, 3 weeks period of time. And afterwards, the clinician will apply the assessment. Based on the assessment results, clinician will be able to calculate the probability, how much the general treatments has works on this specific person. And also, a clinician will be able to calculate the cost of this treatment, how much it costs for the clinician side and the patient side. And also, at the end of the cost, how much the benefit gets, each patient will get the benefits from that kind of treatment. Based on that, clinician will be able to assign if clinician decide based on those evidence, clinician will be able to assign each individual either tier 2, which is more intense treatment, or keep applying the general treatment, which is a PTSD treatment with a really low cost. So the general goal of applying POMD RTI model is that to show that POMD model works better than the shared decision-making process with the low cost to make the veterans to go back their life as soon as possible. And also, POMD model will give a chance to clinicians to forecast their each individual's future situation, and also POMD will give a chance to clinicians to consider each patient's past situation as well to calculate their future situation. This is more detail about the POMD model notations. For example, if you can see here, if each clinician knows what kind of treatments they get, if each patient get, and then what kind of test result they get, they will able to calculate the probability results. And if you see here, C and U, C is the cost function, and the U is the utility function. Also, they're going to be the additional evidence for the clinicians to consider for each individual.
We're going to take a 2 to 3 minute break to fix our audio.
Check 1 2 1 2.
Mic test 1 2. Mic test 1 2 1 2. Test 1 2. Just ours. Test 1 2 1 2. Test 1 2 1 2. It's not very loud at all.
We're good? Give it a test. Figure that. We're just having some weird interference issues, so we're going to move this.
Are you gonna slide it on?
Huh?
So you don't push the mute button.
I want to try to put that down here.
Testing 1 2. Testing house audio 1 2 1 2. Testing house audio 1 2. Testing 1 2. Testing house audio 1 2.
All right. Our wonderful IT people have fixed our sound problem. And ready to go. Ready to go. I appreciate the conversations going on. I hate to break them up, but our wonderful IT people fixed the sound problem. So without further ado, let me introduce Dr. Kun Cheng from UMSL, who's going to talk about discovery of peptide-based checkpoint inhibitors for cancer immunotherapy.
Okay, thank you. So, good morning, everyone. So today I'm going to give a talk about the discovery of peptide-based checkpoint inhibitors for cancer immunotherapy. So I believe everyone here are very familiar with this slide. So last year, our distinguished professor, Dr. George Smith, so he received the Nobel Prize for his discovery, his contribution for the phage display of peptides and antibodies. And since I joined UMKC in 2007, I started working on phage display. So whenever I go out to give a talk, I have to spend a few minutes to explain what is phage display, why phage display is so important. So now with this award, it becomes much easier for me to explain what is phage display. And actually, yeah, thanks, Dr. Smith. And two scientists was awarded this. It's not working. So Dr. Winter shared this award with Dr. Smith for this phage display. And actually, Dr. Winter, he didn't discover the phage display. Instead, he used phage display technology to discover a new drug, which is called Humira. Some of you probably know that. So this drug is a monoclonal antibody, and it's mainly used for the treatment of arthritis. It's very successful. It's one of the most successful drugs in history. So it is the best-selling drug. Last year, the sales for the drug is about $20 billion. So that's why he was also awarded the Nobel Prize for that. So here you can see the importance of this technology. It's a very powerful tool to discover new drugs for different therapeutic therapies. So what is a phage display? So phage basically is a virus. It's a bacterial virus. It only infects bacteria. It does not infect animals or humans, so it's very safe to use. So for each of the phage, you can use genetic engineering to insert a sequence into the genome of the phage. So therefore, on the surface, it can express a protein or a peptide on the surface. The beauty for that is every single phage can express a different peptide or protein sequence. So therefore, you can get a very huge library. You can have about 15 billion different sequences in the library. By using the library, you can screen the proteins or peptides for different purposes. So over the last 12 years, we have been using phage display to identify peptides for ligands for targeted drug delivery. We use that for tumor imaging. And in the last few years, we're focusing on using the phage display to identify peptides for therapeutics, for cancer immunotherapy. So why we're interested in cancer? So many of us may believe cancer only affects a small portion of the people, but actually that's not the case. Roughly 40% of us will develop cancer in our lifetime. So that's a very high percentage, very surprising. And this year, even in the US, about 1.7 people will be diagnosed with cancer and about 600,000 people will die from cancer. So it's a major health problem. And of course, there's so many people working on the cancer research, including us. And in the last few years, the most exciting discovery for cancer therapy is called immunotherapy. It's very successful and very efficient. So what is immunotherapy? As we know, in the body, for example, I'm standing here, I'm very healthy, there's no cancer, but I do have some abnormal cells, or you can call them tumor cells in the body, but I don't have the symptoms. Why? Because I have immune system. The T cells in my body, if there's any abnormal cells, the T cells can find that and destroy that. It can secrete some cytokines to destroy all the tumor cells in the body. So even you have tumor cells in the body, we don't worry about that. But some of the tumor cells are very smart. So they express a protein which is called PD-L1. Once they have this protein expressed, next time when the T cells come to that, the PD-L1 bind to that, and then send a signal to the T cells, don't attack me. So that's how the tumor cells avoid attacking from the T cells, and that's how you get the problem with cancer. The tumor cells keep proliferate in the body and cause many problems. So by understanding the mechanism, it's pretty straightforward to design a drug for that, to block the interaction between the PD-L1 and PD-1, right? So for example, you can design a molecule which is called checkpoint inhibitor. Checkpoint inhibitor bind to PD-L1, and then you can block the interaction between the PD-L1 and the PD-1. And also you can design molecule to block the PD-1. So therefore, in the market, there are 5 antibodies approved to block this pathway. 2 of them bind to PD-1 and 3 of them bind to PD-L1. And these checkpoint inhibitors have been approved for many cancers, and the list will be longer in the future. There's so many clinical trials going on to test the checkpoint inhibitor for different cancers. So we have been working on peptide-based checkpoint inhibitors because we believe peptide are advantages over antibody because they are very small, it's easy to make, and a lot of the major advantages is they have better tumor penetration, and also it's much easier to make bispecific inhibitors. So why we believe peptide has better tumor penetration? The difference between the antibody and the peptide is like the antibody and ant. Well, the antibody is about 1,000 times bigger than the peptide. So therefore, we believe that small peptide have much better tumor penetration. So why tumor penetration is so important? So let's look at the tumor. In the cancer patient, tumor is not just a single cell. There may be 1,000 or million cells grouped together form a 3D structure. So for the treatment purpose, you have to deliver the antigen or ant into inside of the tumor, not just on the surface. But antigen is too big, as we know that. It can only arrive on the surface. It can only block the pathway on the surface. It cannot penetrate into the tumor. But if you have a peptide, because of its small size, it can penetrate all the way into the tumor to block the checkpoint inhibitors inside the tumor. So we believe the peptide will have better effect compared to monoclonal antibodies. And secondly, people now are interested in combine different antibodies together to target different pathways at the same time. One endpoint is too big. If you have two endpoints, that's too big. It's not easy to combine 2 or 3 endpoints together. But if you have peptide, it's pretty easy to combine 2 peptides together. And if you want, you can actually attach more. You can attach 5 or 6, whatever, to form a combination therapy. So that's the reason we work on peptide. So we use phage display, discover several peptides, and then we look at the activity in a tumor model. For the mouse, we implant tumor cells here. And then we give the antibody and give the peptide for several times to look at the tumor size. So here are the results. And as you can see here, for the untreated group, you get pretty big tumors there. But for the antibody, it shows pretty good effect. You can see there, most of them show very small tumor size. We have 3 peptides. The first peptide 1 didn't show any effect, but peptide 2 showed pretty good effect. It's even better than antibody. And actually for 3 mice, we cannot find the tumor after the treatment. So here are the conclusions. Phage display can be used to discover peptide-based checkpoint inhibitors. And we found one peptide which shows very good anti-tumor effect. And so it's very promising checkpoint inhibitors for cancer immunotherapy. So we are in the process to file a patent, and we also got a grant from NIH to support the research. And finally, I'd like to thank my postdoc and student working on this project, my collaborator from MU School of Medicine, and also financial support from NIH and American Society. Thank you so much.
Next, I'd like to introduce Dr. Anthony Convertine from Missouri S&T. His talk will be Polymerizable Prodrugs, New Tricks for Old Drugs. We'll figure it out.
Control L.
We have.
Say Control L.
Control L is
Control L. Well,
Perfect. All right, I'd like to thank the organizers for giving me this opportunity to speak today. As you mentioned, I am from Missouri S&T, and we are working within the Center for Biomedical Engineering here. So our research is focused on nanomaterial synthesis. We do a lot of macromolecular synthesis, a lot of nanoparticle synthesis, and we're doing this for a wide range of therapeutic applications. We've got projects working to make neuroprotective materials for traumatic brain injury. We've also developed materials to deliver biologic drugs such as short interfering RNA, messenger RNA, and proteins into the cells, and that research has actually gone all the way through to non-human primates. But as the title of my talk suggests today, I'm going to talk about polymerizable prodrugs and how they can teach old drugs new tricks. And we've worked on this for infectious disease applications as well as cancer chemotherapy. So as the title of this slide says, we don't want to make new drugs. We want to make new drugs better. The cost of bringing a new drug to market, depending on the source you look at, is between $650 million and $2.7 billion. And all the really easy to hit low-hanging fruit molecular targets have mostly been exploited. These new targets and old drugs often show very poor pharmacokinetics, pharmacodynamics, and toxicity. In order to address these challenges, the field of drug delivery has published literally tens of thousands of papers and almost as many patents on micelles and liposomes and other ways of encapsulating drugs. But these studies and all this work has really resulted in very few products actually reaching clinical trials beyond the occasional phase 1 trial. In contrast, polymer drug conjugates, where the drug is reversibly attached to a macromolecule scaffold, have a number of drugs in phase 2 and phase 3 trials. But despite the promise of this, these materials are still complicated to make, require multi-step syntheses, and are not really very scalable. To get around this, we've developed what we're calling drugomers. So we attach pretty much any drug you want to a polymerizable group via degradable linkage, and we can control this linkage. All the synthesis can be done in GMP laboratories, giving you a pharmaceutically pure compound. And then a single step, we polymerize it all together with targeting moieties and molecules to solubilize everything. So the project I'm going to talk to you about quickly is one we did for the Defense Threat Reduction Agency. They were interested in combating pathogens that the Russians developed during the Cold War, specifically tularemia and melioidosis. These are extremely lethal pathogens and have greater than 50% fatality, even with current standard of care. And that standard of care is generally IV bolus medications as well as oral medications and extended. And these treatment regimens are pretty toxic and very expensive. Also, antibiotics are a hot area because, as the director of CDC said, we're entering the post-antibiotic era. So, quickly show we can make these things with very well-controlled zero-order release kinetics. It's very linear over anywhere from a few hours to several days. The proof is kind of in the pudding. So what we did is a aerosolized infection model, and you can see even with the free drug, this is 100% lethal model, and Ollie almost died just after 6 to 7 days, whereas we were able to recover 100% of the animals to the end of our study. So we're looking at S&T to integrate these things into advanced materials for wound repair, bone recovery, and we can just simply polymerize them into whatever scaffold you want using off-the-shelf 3D printing technologies. Now, we've talked about antibiotics, but we've developed these materials for HIV and anti-malarials for the Gates Foundation. So any drug you might be interested in delivering, we can integrate these into materials that quickly and easily dissolve and can be rapidly moved towards clinical applications where clinicians can just use the material. They don't have to formulate it, figure out how to dissolve it or use it. We also have a lot of work ongoing in traumatic brain injury to develop materials that neutralize the secondary derangements associated with the issues. I know there's a lot of people working in that area, so I just thought I'd stick a slide in right at the end. I'd like to thank everyone for the opportunity to speak.
Our next presenter is Raghuram Kannan from the University of Missouri, Columbia. The talk is Design and Development of Nanomedicine Drugs and Metal-Based Radioactive Pharmaceuticals.
Well, thank you very much. The title was more, it's longer than 5 minutes, so I cut it short into cancer nanotechnology. Thank you very much. I am Raghu Kannan. I'm a faculty in Department of Radiology and Bioengineering at the University of Missouri, Columbia. Our group is focused on utilizing nanotechnology-based tools to understand cancer. So what do? We go from one extreme of cancer part to the other one. We try to understand the mechanism of the proliferation and metastasis of cancer, try to develop the drug to kill the cancer. So what do we do generally? So we work mainly on three aspects in the cancer. We try to identify the targets, what we call it as a genes and markers, which are responsible for the cancer growth. The cancer, especially what we use is, or what we focus on drug-resistant cancer, not the normal one, the one that villain comes back and hit at patients. So we try to understand what genes or what villain is making the cancer to grow or not respond to the drug, and then try to use a gene therapy to overcome that. And we also work on something called an imaging device. I thought I'll put that one too. We have a long list of collaborators who work with us. Some of that not related to cancer, some of that are related to cancer. We have outside institutions which are working on that in parallel with us, where we run our some level of clinical trials. In the case of NIR imaging device, what we try to do is we try to see the fluorescence inside the body. So we developed our own device. The idea is to see the cancer with precise margins. That is the whole idea of starting this part. The one that I am interested in forming collaboration and moving forward is like we are trying to develop a UO1 and a /and RO1 type of a grant on this is the gene therapy, which we call it as an RNAi. RNAi is you are cutting down the drug resistant gene. or you are controlling the volume of the gene by silencing it. You are not removing it like CRISPR. You are silencing it so that side effects are low. Let me take one example and walk you through what we have done so far. What we have shown here is non-small lung cancer. The lung cancer affects a lot of people. In Missouri, a lot of people die out of lung cancer. The nationwide, worldwide, lung cancer is still a major issue. The problem is, or let us go to that. When the patient comes into the hospital, you take a small amount of the tissue, analyze through the biopsy, what are the driver genes that is responsible for the cancer growth. Let us say in this slide, we have put EGFR mutant, EGFR positive. The patient is EGFR positive. The oncologist comes back and says, take erlotinib. Exactly within 6-7 months, the patient is cancer-free. No worries. 1 year, you should see the image of the patient. 0, no cancer cell at all. But there is a villain, super villain, which we can call a drug-resistant gene that is still present in the body. Okay, that comes back and hits the patient very hard, and the patient succumbs to the disease. What is that drug-resistant gene? Why it happens? What is the mechanism behind it? That is the focus of our research. Okay, let me try to give you an idea how we do it. We start the patient. We do a trial in taking the biopsy of the sample, do an RNA-seq, try to identify the gene, and follow up the patient when they get at FU1 and FU2. Try to do the gene. I have not even started. So the FU1 and FU2 at the stage where we know that like there are some unique genes, driver genes, which we call, is responsible for the cancer growth. Okay, this is the gene which we go through line by line out of this 1041 and try to understand what are the genes that is responsible for this cancer growth. Can I come back and combat it? Yes, we do. We try to do a PCR. In the particular work, we identified 2 genes, AXL and FN14. I'll come back to that in one slide more. And then we do a Western to identify the protein. Protein is an action point. We do proteomics to identify and characterize the protein that is responsible for the gene. And we do an IPA network analysis. What do? We try to understand the mechanism, how it became resistant. Just to giving an idea, in this particular work, we identified a crosstalk between the AXL and FN14. To the best of our knowledge, this is the first time anybody has reported a crosstalk between the 2 genes in a lung cancer, why it became drug resistant. So we didn't stop at that stage. We went and designed a nanoparticle which can deliver 2 genes. Okay? What is the big thing about it? If you put an RNA as such, what you can see here, it will die on its own. Within 10 seconds to 20 seconds, RNA will disintegrate into your blood. By putting into our nanoparticle, we were able to keep it for 48 hours. So we were able to convert that, which is drug resistant at 40 to 8 now. So it is going to work. So we are making the existing drug to work much better. Are we just saying it? No, we did that in the mice model. Okay, the idea here in this slide is not to focus on the image, but focus on the rightmost part of the graph here. The AXL and FN14 is in the negative. What I am showing here is a protein. We were able to downregulate the protein that is responsible for the drug resistance. So we didn't stop at that stage. We did an animal model. Like I'm going to put all 3 together so that we can cover some time here. We did multiple animal models to show that like our drug resistant cancer can be reversed or maybe more responsive for the existing drug. If you can solve that, then there is no problem of cancer by knowing which gene is affecting the patient. If you can see these graphs, the downmost, bottommost part, that is a tumor growth. You know, like when I suppress both the genes together, okay, then the existing drug, which is resistant now, start responding. So what I'm trying to do here, using RNAi to identify the gene. By knowing which gene is problem, I kind of control the volume of the gene and make the existing gene to work. This can be expanded to any number of cancers of our choice. This is the major issue for cancer. Primary cancers are solvable with surgery, and we have a lot of good drugs. Immunotherapy is a great part to go with, but when it becomes resistant, there is no option. So you have to know the gene and control the gene, and you don't want a long-lasting effect. So RNAi is the area to temporarily suspend the gene and make your resistance to work with. And we are looking for collaborations on this area. If you're interested, I have my email down, or you have it in this. Just let us know. We'll be happy to work with you. Thank you.
Next presenter is Julie Seaman from Missouri S&T. The title of her talk is Biomaterials and Stem Cells in Precision Medicine.
Thanks to Costas and Jennifer Pratt for organizing this today, and thanks for everybody for coming down to Rolla today. So I think it's probably been more than 1 to 2 years since most of you guys took a biology class. So just a real quick pop quiz. How many different types of cells do you have in your body? Not total number of cells, just types of cells. 2, 20, 200, 2,000, 2 million. I'll buy you a cup of coffee in the back. 200, yeah, so about 200 different cells in your body. So for those that's been a while since biology, those 200 cells, they're outside of what we call the cell cycle. So the cell cycle is where the cell naturally divides. So if you remember the terms mitosis, meiosis, anaphase, prophase, those are all the buzz terms for the cell cycle. There's the Science Center for those in St. Louis actually has a good exhibit this month if you guys want to refresh yourself. So if most of the cells are out of the cell cycle, they never divide, then what happens to our damaged or our diseased cells? So it's been a terrible, terrible winter, and now a lot of us have been outside. We've probably got sunburn, so we've damaged those cells. We know that they're replenishing some from somewhere, from somehow, but once again, it can't be their own cells since they don't actually divide. Anybody have any idea where the miracle cells are coming from? The hints is on the board. Stem cells. And so that's what we do. We work with 2 different types of stem cells. We work with stem cells from bone marrow as well as from adipose tissue, which is a fancy word of saying fat. So we've shown, and so just like in the last talk, Dr. Shoup was talking about in the clinical trials, there's a lot of good evidence out there that these stem cells are working. They're working great in our laboratory animal experiments, but a lot of the clinical trials are just not working that well. And so we're trying to figure out why. We're trying to figure out who has actually good stem cells versus whose stem cells need a little extra help. And so it turns out if you're over the age of 60, if you're diabetic, which is about 14% of the population of Missouri, if you have an autoimmune disease, neurodegenerative disease, these stem cells that replace all the other cells and that help the cells, they're not working as well. And so we've shown that in a lot of different models. We've shown that with disease stem cells from animals. We've shown it from older people and from humans into animals. And once again, it's shown that it's not working. But the clinicians aren't listening to us fast enough. And so in the clinic, they still want you to use your own cells, which we're kind of getting from a different type of cell, where you have to match your blood type. If everybody knows if they're A positive, B plus, that. So we're taking that knowledge and applying it to our different cells. And so if they're not listening to us, then we want to try and come up with a way to reboot these cells. So this right here is borate bioactive glass, which was invented by Dr. Delverde, who's sitting back here and can answer more questions about the development of it, and his laboratory. And he's using it to treat wounds that don't naturally heal on their own. So this would be very important for, once again, people over the age of 65, as well as diabetics. And so the first couple pictures right there are exactly, looks like we don't have our little pointer, but the 2 columns on the left are both diabetic patients. So diabetic, once again, you have defunct ASCs, the adipose stem cells, they should naturally heal. And then we're missing a little picture of the dog that resolved, but he resolved within 20 days. And these guys, what's really cool about this is these guys are healing without scars. So there's only one population out there that heals without scars. Anybody know who that is? It's babies still in the belly. So if you do in utero surgery, typically you don't have scars. Once you come out of your mom, you have scars. So if you look at your scars, usually the skin's discolored, usually you don't have freckles, usually you don't have hair. But if the picture of the dog, maybe he's next. Yeah, there he is. So you can see that the hair is regrowing. And if you pet him, he actually can wiggle around. There's a nice video of a horse too that you can see that has nerve sensation back. So it's healing all of these things when it shouldn't be healing. And so for us, coming from an adipose stem cell area, we know that the adipose stem cells has a strong role in each one of the steps of wound healing. And so naturally, we're thinking that this glass is actually affecting the stem cells of people who have defunct stem cells. And so potentially, this is a mechanism that can reboot these stem cells to make them happy, healthy cells. So we've done that. We've done some molecular assays, which we don't have time to address them here. But if you guys want to know more about the assays or the techniques we use, or if you want to see the lab before you guys leave Rolla, let me know. But they do change the cells. And so now we're getting cells. We have cells from diabetic patients. It's harder to get MS patients up here in Missouri. But but it does, with our preliminary results, does look like that these cells will change these disease stem cells into happy, healthy cells, hopefully changing the prognosis in the clinic. Then that's what we do, and this is who we are and our collaborators. Thank you very much.
Next presenter is Krishna Kolan from Missouri S&T. His talk is novel method to 3D bioprint stem cells with composite bioink.
Good morning. My name is Krishna Kolan. I'm a postdoc working in the Department of Mechanical and Aerospace Engineering. My faculty advisor is Minglu. So we are from the additive manufacturing lab. We work with different 3D printing processes, metals, polymers, ceramics. But in this talk, we'll focus on using bioprinting. So just a brief introduction. Bioprinting, most of you must be known, So it's basically we take cells and we mix it with some kind of hydrogels, or it could be other materials along with hydrogels. So we deposit them in a layer by layer structure, and we get this tissue and we mature the tissue in incubator for maybe a month, depending on the type of tissue, and we get the tissue there. So what are the different applications in bioprinting? You can use it for tissue engineering, regenerative medicine. We can directly, for implantation or tissue repair, or we can do drug discovery or high throughput assays, or we can use it for cancer research. For example, we take cancer cells, mix it with hydrogel and make a 3D model, and then use it for drug discovery. So there are three important aspects in bioprinting. So one is the materials, the fabrication process itself, and then cells. So in materials, Julie just talked about the borate bioactive glass. We have Mosaic Corporation and today. So we use bioactive glass, borate bioactive glass. And for the cells, we use adipose stem cells with Julie in her lab. And then the process, the commercial bioprinting processes are mostly melt deposition bioprinting, where we use heat to melt the polymer. But the problem is it has high temperature, so we use a novel process called solvent-based bioprinting. So what we do is we take bioactive glass, mix it with the polymer, make a paste, extrude it, we can print it, and make any kind of shapes. In the pictures you see, let's say, a form of a nose, half-built nose. And then the microstructures, what we basically study is how the glass is being released from the polymer matrix. So unlike the commercial bioprinters, we can see here we can use different polymers and control the rate of glass dissolution from the polymer matrix. We can use PLA or PCL and control the degradation. We can also use different bioactive glasses. So we tested that in animals, and we found, you can see that the bottom picture shows a cranial histology of a cranial defect in a rat. So you can see that when we use a PLA matrix instead of PCL matrix, the borate glass dissolves much faster and there is more bone growth. So this is a short video of the bioprinting. So we have two different syringes. In one syringe, we use bioactive glass and polymer matrix. In the second syringe, there is bioink. It's alginate gelatin-based. We mix stem cells in that. So it is a deposition process in a layer by layer fashion. And towards the end, you can see it starts to form. We can do it in different shapes. This is just a testing phase, so you can see simple square shape. So what we have come to a point where we found out that alginate and gelatin composition works best, and then we did incubate the scaffolds with cells in them for 7 days or 14 days, and then I think it's kind of difficult to see in the picture, but we see a good viability. Immediately after printing, we get more than 80% viable stem cells in there, and then after 7 days and 2 weeks, we get like 70% viability. there are two different ways in which we can add bioactive glass. That's what we are working. So first thing is, as I said, we mix the glass in polymer matrix, but there is a second way where we can directly add glass to the hydrogel itself with cells. So in that process, we see that there is initial pH change and shock where there is cell death immediately after printing, but they seem to be recovering and do very well after 7 days and 14 days. So what we try to do in future, or the future directions, or for collaborations, is that now that we have come to a point where we can mix cells and do this 3D models, the idea is that here it's a just example of how we can make a vascularized in vitro bone model. So what we can do here is we can use stem cells or bone cells and print a model in a layer by layer fashion where we can perfuse endothelial cells, make them as vascular structures. So the idea is once we develop this 3D model, we can do drug testing, we can add different drugs and see the effects of drugs on the cells, or we can even study effects of different bioactive glasses. We have a good glass ceramic engineering program here at S&T. Or we can also do cancer research. We can put in cancer cells and for example, breast cancer cells and bone cells, and study cancer metastasis, things like that. We can do a lot of different analysis using these 3D models. So thank you, and we have our contact information if you'd like to.
Thank you. The next presenter is Dr. Stefan Lofeld from UMKC.
Hello. I'm glad to be here. Probably everybody in the biomedical engineering business has heard about or even seen scaffolds for tissue engineering made via additive manufacturing processes, also known as rapid prototyping or 3D printing. And if not until like 3 minutes ago, thanks to Krishna, now we're introduced to them. So these processes allow to make very complex structures, and depending on the process that you choose and the resolution you can achieve with them, you will utilize them to make different models. And as we said, we also use them to make scaffolds for tissue engineering in cases where we just need a support for anything we want to implant, or where we also need to provide a three-dimensional structure to tell or to let the cells know in which direction to go for critical size defects. So depending on what you want to achieve, you might choose one or the other additive manufacturing process. I use what I had available in some of my previous research, and I use selective laser sintering, where a laser is used to heat and fuse powder particles in a powder bed together to form a three-dimensional structure. In this case, we used a polycaprolactone polymer, and we added 10% true calcium phosphate to that material to enhance bone ingrowth into the scaffolds. We wanted to compare these novel scaffolds to the gold standard in bone tissue engineering, which is a pure true calcium phosphate scaffold, to overcome some issues with this material. For instance, its brittleness, that you have no control about the porosity, or size, or interconnectivity, and so on. So these two scaffolds were implanted into sheep tibia and let in there for about 14 weeks, up to 14 weeks. After excising them, we saw results like this, and you would suggest from these pictures that the TCP scaffold is still significantly more successful than the polymer scaffold we use. Now, as you probably know, in X-rays, calcium phosphate shows up almost similar to bone because of the similar density. So from these pictures alone, we cannot tell if we actually had bone ingrowth into our TCP scaffold, while for the polymer scaffold on the right side, it just looks like a void. But that's just due to the low density of the polymer, so it doesn't show up. The micro CT scan then actually told us that we do have some bone ingrowth on the right-hand side in our polymer scaffold as well. So this looks like scaffold material, but actually it's the bone that left gaps where still polymer is in place. Nevertheless, the TCP scaffold was better. I have to admit that. The PCR material is probably not the best for this type of bone formation. And but we proved the concept that we can process this material and add the second phase with selective laser sintering. And actually during the process, or during the duration of the project, we managed to introduce up to 50% tricalcium phosphate. And this second phase changes the entire production process, all the parameters that you have to see. So it's not that easy. Unfortunately, we couldn't do another animal trial due to funding and duration of the project, do another animal trial, but we would have thought that this would have performed much better in that trial. So far for the process, let's get to the materials, or rather the shape of the materials. I think microspheres are very good to utilize in scaffold for tissue engineering. So when packed together, these microspheres to form a scaffold, they have an inherent porosity due to the gaps between the spheres. The manufacturing process of the microspheres allows us to encapsulate growth factors, secondary phases of the polymers, drugs, or even cells. And by the biodegradation, we can actually release those drugs or secondary phases in a timely manner after implantation of the scaffold. Also, when you use shell core materials like different polymers on the outside and inside, you can tweak the mechanical properties of the scaffold you're trying to make. And sintering with subcritical CO2 has shown not to harm any cells that were either encapsulated in the microspheres or were seeded onto the scaffold before sintering. By filling molds with two different types of microspheres, you can also create gradient scaffolds when you change the ratio of your two materials, as you can see here in the red and white scaffold. However, there are also some disadvantages. We do have a porosity, however, we have no big control over it. So we cannot say where we want to have the pores, we have no control over the shape of the pores, and the size is mainly determined by the size of the microspheres. Also, we have no inner architecture, we can only choose a mold to give it an outer shape. And in gradient or multi-material scaffolds, you have only little control over the placement of the second phase. So you have a gradient, yes, but you cannot particularly place different phases at different sites within the scaffold. So the idea was to create, to combine 3D printing with the microspheres. And to be able to print the microspheres, I had to use a binder, a matrix material, to keep the microspheres in shape until they're fully sintered and stick together. So I started off with a very simple 3D printing process, homemade type, and a small range of different matrix materials. And with them, I was able actually to create strands of microspheres that was able to be extruded from a syringe and that stayed in shape until sintered by the high viscosity that I chose with my binder. Now, with this system, I couldn't create very high pressures, so I couldn't go down to very small diameters. Now, fortunately, the School of Dentistry at UMKC has recently invested into a high-end bioplotter. So with this one, I have higher pressures for printing. I have an accuracy of up to, or a resolution in printing of up to 1 micron. So I can really focus on certain areas within a scaffold, and I can use up to 5 materials for the same scaffold. So I can really play around with different materials at the core of the scaffold and at the outer side. So by combining the 3D printing with additive manufacturing with the microspheres, we can add drugs and growth factors at specific sites in the scaffold, and that will allow us to release those different types at predetermined times after implantation to support tissue growth, to supply nutrients, to counteract tissue reactions, and so on. We can also apply gradients of materials and mechanical properties within the scaffold, and we hope to create better scaffolds, for instance, in areas where you have bone tissue and cartilage tissue interfaces, so in joints. We can also add stiff load transfer tracks to a softer scaffold, where the softer material is actually necessary to enhance bone growth or tissue growth, while you still need to provide a track for the loads. And we can apply an architecture to optimize load transfers throughout the scaffold and play around with the porosity to enhance cell attachment and growth. So the possibilities seem endless, and I'm open to suggestions and collaborations with this. If you're interested in 3D printing, why not come to our symposium in Kansas City in June this year? Registration is open under mr3ds.com. Thank you very much.
Next presenter is Brett Ullery from University of Missouri in Columbia. His talk is Pharmaceutical Formulations for Pets and People. My two favorite things.
Me too. So thank you all for the organizers for setting this up. It's exciting to be here. As mentioned, I'm a faculty member in engineering from the University of Missouri Columbia campus. So you may have heard this phrase of all politics is local. I think all research is really local too. So my family is made of our two dogs, Colby and Jack. And my wife, Dr. Ullery, the real doctor in the family, she's a veterinarian and a clinical instructor in the vet school at the University of Missouri. And so, you know, probably like many of you, your families exist of both pets and people. And so I want to make a difference in both of their lives. And so while maybe I'm a little biased because I'm married to a veterinarian, I think about clinical applications when it comes to human and animal health. But we can think about that there are many opportunities that are available here, even if your focus is primarily on human health. First of all, we have to do preclinical studies. You've seen many of them in the biomaterials session here today, right? Of things that we need to do with small animal or even large animal studies to eventually get to doing human trials and human clinical medicine. So if we think about not just doing that as a kind of a stepping stone, but an opportunity to get into companion animal and production animal health, there's a lot of options that are available. First is that the regulatory hurdles are lower, so it's an easier pathway to get to clinical market. And vets, they, again, maybe a little biased, but they like to see new products get in and make a difference in their patients. And so they're very early adopters. Also, while it's not as lucrative of a market as you would see in human medicine, it's still a pretty large market. So there's a huge opportunity for us to kind of get over that valley of death that happens with biomedical products by creating revenue streams. So I'm a biomaterials researcher, so it's kind of been fun to be at the end of the session to kind of you know, maybe bring it home here at the end of opportunities that we can use as many different materials that have been proposed here, right? Things that are ceramics, metals, polymers, composites of those. And so where biomaterials has kind of, you know, reached a new plateau is, well, how do we get to that next step? How do we have new engineered solutions? So, you know, the materials that are out there have done a really good job for a lot of applications, especially in the biomechanics field, but how do we design things for really complex strategies, things like intracellular drug delivery for cancer therapy, which was talked about earlier, or tissue engineering, complex tissue engineering that has been brought up in a couple of different talks here. And so what we think about is kind of a sub-area of biomaterials called biomodulatory materials, which actually you've heard a lot of today, unlike Dr. Convertine and Dr. Seaman, you know, these materials that then are able to actually have physicochemical properties that induce different types of biomedical outcomes. So I'm going to just tell a couple of quick vignettes about the research that we're doing in one of the areas. So the first is to talk about blood cancers. So these are hematological cancers like lymphoma and leukemia. These affect about 500,000 patients per year in human medicine, but also in animal medicine. Lymphoma is actually the most deadly dog cancer. So if your dog lives long enough, it's probably going to die from lymphoma, unfortunately. So we have this opportunity at this interface to make a difference. So our collaborator, Mark Daniels in the immunology department at the University of Missouri has found a peptide inhibitor that has really broad spectrum activity against a number of lymphomas and leukemias. So that's really exciting, but you know, does that just have like a chemotoxic effect, or does it actually kill the cells of interest? And then what happens to healthy cells? And so if we look at this, luckily there isn't really a response that we see in killing healthy cells. So that's great, but the problem is a peptide drug by itself can be pretty limiting on its ability to kill cells. So he was very interested in kind of having some sort of drug delivery solution. So what we do is we make lipidated peptides. So by adding this lipid region, we actually get self-assembly into different nanoparticles. And those nanoparticles for this application are about 10 to 20 nanometers in size. So good for trafficking in the blood and getting to the cells of interest. So what we see is comparatively to the standard peptide therapy, our nanoparticle therapy actually helps pretty significantly. We've started to work in aptamer targeting with Donald Burke, who's an RNA biochemist, and then collaborations both in the veterinary and human medicine side, and eventually wanting to go into looking at cancer vaccines, so actually being able to help tune the immune response to prevent cancer relapse in the future, which was kind of mentioned with some of the PD-1 checkpoint inhibitors. So there's a lot of opportunities of collaboration I can really see coming out here. So we also have the opportunity to actually just use these as vaccines. That's where the micelle field kind of started, at least in my research. So if we go ahead and vaccinate animals, it's interesting that structure plays a really important role in the type of output response we get. So if we have kind of small particles, they actually enhance the vaccine. But if we have large particles, these blades, we can actually diminish that. So for different applications, we have a real opportunity here. And we've started to work with a number of different people at the University of Missouri, including computational research, to help us find new peptide antigens and vaccines that then we can modify and evaluate both in animal health and in human health. So Dr. Stitch works in Lyme disease, so we're thinking about like reservoir and animal reservoir, and Dr. Hom works in influenza, so trying to develop new universal influenza vaccines. So what's the advantage of being a part of the system? I think it's been kind of hammered pretty well so far, but we have a lot of colleges at our individual universities, so a lot of expertise to get to work together. Every school, every campus has an arts and science campus, lots of engineering. And as mentioned, we've got dentistry, pharmacy, veterinary medicine, human medicine. So those opportunities to work together. And one of the things that brought me to the University of Missouri Columbia, and that's expanded out as I've met researchers at a lot of other universities, is that everybody's passionate about this transdisciplinary research and working together. There's not a lot of big heads out there. I feel like everybody likes working together. And I think that's coming together for the system with the TPMC, which is being built at the Columbia campus. And I think that's a real opportunity for all system researchers interested in the biomedical space to work together. So thank you.
We're going to move to a slightly different topic, the topic of biomechanics. The first presenter is Trent Guess from Missouri and Columbia. The title of his presentation is ACL Injury onto the Knee, Mechanisms, Prevention, and Treatment.
Okay, before I give a talk on knee biomechanics, I typically ask my audience, who in here has torn their ACL? Usually get a few hands go up. Then when I ask, all right, who has a friend or a teammate has torn their ACL? That's when I see most of the hands in the room go up. So ACL injury is common. If you are a female high school athlete who participates in both soccer and basketball, and in any given year, you have a 5% chance of tearing your ACL. So ACL injury is not just devastating to the individual, but it's also, when you factor in disability, societal costs are greater than $10 billion per year. And even with our best efforts to treat ACL injury, chronic knee dysfunction persists, your chance of re-injury increases significantly, and I think most importantly, is that if you tear your ACL, even if you have a reconstruction, you have a greater than 50% chance of getting early onset osteoarthritis. So preventing ACL injury in the first place should be our primary concern. But to do that, we have to understand the mechanisms of ACL injury. So most ACL injuries are non-contact, in fact, the vast majority. And they occur when an athlete plants their foot and then they tries to cut or turn. And the ACL is strong. It takes about 450 pounds of force to tear an ACL. So the question is, how in the world do we tear an ACL just by jumping and cutting? Well, we are combining motion capture data with medical images and computational methods to predict the loading on the ACL. We have developed musculoskeletal models at the body level that include knee tissues, such as the menisci, ligaments, and cartilage. And you can see, like during normal cutting that we're simulating here, ACL forces are actually quite small. They're on the order of walking. So, when you tear your ACL or you look at videos, usually there's some kind of disturbance. There's a defender that jumps close to athletic, there's some disturbance, and so we know that neuromuscular control plays a key role in ACL injury. We can simulate changes to motor control, muscle activations in our models. We know that, for example, calf muscles are involved in turning and cutting. When we increase the neural command to the calf muscles during our cutting maneuver, we see significant increases in ACL force. We also see body positions, knee positions, signatures in our model that match what we know about ACL injury. We see body positions where the ACL is vulnerable to injury. We see at max ACL loading positions of the tibia femur that match the positions of the tibia femur at injury that we know from bone bruises. We know that when you tear your ACL, more often than not, you also injure your meniscus. Most of those meniscal injuries occur in the posterior region of the meniscus, and that's exactly where we see pinching of the meniscus in our simulations. We also know that there are anatomical risk factors. For example, a lax knee, lax ligaments, posterior tibial slope, a shallow tibial plateau, all make it easier for the femur to slide relative to the tibia. So in summary, when we have a vulnerable position, or when we're in a vulnerable position, we add excessive calf muscle forces, we push the tibia forward, pull the femur back, pinch the meniscus, and put loading on our ACL. So how are we using this information to help prevent ACL injuries? Well, we're working with sports medicine physicians, physical therapists to develop new methods for assessing young athletes. We're using the information that we're getting from our computational models to inform these assessments. For example, we're using depth cameras, portable force plates to screen for faulty movement patterns in athletes, faulty movement patterns that are associated with increased ACL injury risk. We're developing new technologies to measure tibiofemoral motion. That allows us to come up with new methods for screening, diagnosing, and assessing knee function. So essentially, with the help of our computational models, our team of engineers and clinicians are kind of changing how we view ACL injury and how we prevent and treat ACL injury. Thank you.
The next presenter is Dr. Yun-Sung Song from Missouri S&T. He's going to talk about biomechanics and mechanical engineering.
Okay. Yeah, hello. My name is Yun-Sung Song. I'm from Missouri S&T. I'm one of the 6 speakers of the department of the school. And I think I'm one of the 2 mechanical engineer in here, and then I come with the shortest title at the top, I think. Biomechanics and mechanical engineering. Yeah, I want to start my talk with talking about scales here, because in many cases, biomedical engineering is viewed as a molecular level, chemical level, atomic level engineering. Whereas I come with another realm at a totally different length scale of meters, like velocity tens of meters per second, large masses with our body parts, and forces up to like 1,000 or 2,000 newtons, and then energy exchange in kilojoules. Those can happen in during physical therapy, for example, in healthcare as well. When you work with body parts, when you want to rehabilitate the function of the body parts, physical therapists work together with the patients, and the interaction between the patient and 2 agents here, the patient and the therapist, can lead up to this length scale, this force scale, this energy scale. So the classical mechanical equations like F equals ma, like F equals kx, the energy being mgh, right? Those kind of things can still apply even though they seem classical. There are implications of those classical mechanics in healthcare as well. And it turns out the mechanical engineers are one of the people that have been working on those length scales for some period of time. So for example, let's see what are the big length scales that we are, as a mechanical engineers, interested in healthcare. And one of the big examples is stair negotiation. If you go up the stairs, like say 1 story, you have to consume about 2 kilojoules. That's just counting your body weight displacing about 2 meters in height. Right? Your leg muscles have to generate all that work. 2 kilojoules don't come from anywhere else but your leg muscles. So 2 kilojoules you have to create. On the other hand, when you're coming down the steps, you have less of a potential energy. Where do they go? Right? There's only one way for those energy to go. It has to dissipate from your muscles. Your muscles have to do exactly the same amount of energy generated, but it has to be negative work. So one is positive, one is negative work. That's kind of fundamental mechanics here going on. And that suggests that going up the stairs and going down the stairs, they could be equally demanding in terms of muscle energy demand. Right? And if we can help going up or going down the steps, we can help people go up the steps, and we can help people go down the steps, and both of them can have as much impact on the stair negotiation. So here, yes, that's just a lump parameter, simple classical mechanics representation of what I mean by positive and negative work. So, for example, we can test the idea. We can help the stair negotiation both going up, down, or going up by harvesting some of the mechanical energy, and it's not some because it's about in the order of kilojoules. So going down the steps becomes easier. Going up the steps becomes easier. And I have I didn't include the results slide because they're rather busy. It's sufficient to say that this worked to reduce the knee joint work by about 30%. Another example that I want to bring up is falls. And then this is one of the most prominent life-threatening physical injury in older adults that leads to reduced mobility, typically leads to more reliance on the assistive devices such as the cane here. My motivation is, okay, the cane gives different aspects of support. Can we harness some of them, only some of them, and provide some targeted balance assistance for those who don't actually need the physical support? So the big question is, can we even provide balance assistance even without the physical support? We just cut the physical support, provide sensory information, does it help? We developed what's called the virtual cane. So it feels like a cane, but it doesn't touch the ground, but it measures the distance to the ground and feeds back the information from it. Turns out that it gives as much balance assistance as the physical cane does. So what's going on there, right? It's the sensory information that is being fed into the patient that is, well, in this case, it's a healthy young, but it's helping with the standing balance. So that's also another element of large-scale movement here. The last thing I want to talk about is between human and human interaction, or between human and robot. If we're helping each other move across the room, for example, holding hand in hand, what should we do? Well, how should we build a robot to do that? And the simple question of what's the scale of the force between the two humans is rather unknown. So the literature on the human-human interaction down in the red square there suggests that there are only 2 or 3 studies, but it suggests that the interaction force between humans could be low when one human is assisting another human to walk. Now that's a little puzzling, because if you want to assist another person, and then you want to have a firm grip between the two persons, that firm grip rather prevents you from exchanging information through forces, and this is assuming the forces encode information, because if you're having a large grip force, then it makes it difficult for you to detect small forces, unless your arms are doing something else, where your arms are more compliant and you can detect length changes much easier than you detect the force changes in your hand. And that's a big hypothesis, but if that hypothesis holds, then you can change all our aspects about the design of the robot, and then how we approach the physical therapy between human therapists and a robot. So here I showed, this is my last slide showing the outline of one of the most recently funded projects to uncover the human-human interaction, and by looking at the basic classical mechanical parameters like the forces, the displacement, the energy exchange, and how to use that knowledge to better improve the human-human interaction and also the human-robot interaction. And as an engineer, I'm also interested in building this robot and using it as a research tool as well. So for most of the talks here, we're talking about drugs and chemicals. What I want to say as a last closing comment is that these interventions that coming from outside, like mechanical interventions, don't have to, they can actually augment the therapies coming from the chemicals, right? They're not exclusive. They're probably augmented to each other. So I would like to pursue the opportunity of using some of the classical mechanics into the low length scale molecule therapy as well. So, thank you.
The next presenter is Dr. Ganesh Tyagarajan from UMKC. The title of his talk is Role of Mechanical Strain in Bone Forming Pathway Activation in Osteocytes, in vivo and in silico studies. Where's the presentation?
Thank you to the organizers for this wonderful research symposium. My name is Ganesh. I'm from UMKC. I'm a civil engineer, but I've been collaborating with the School of Dentistry. I'm from the School of Computing and Engineering, with bone biologists and also dentists working in the bone areas for almost 15 years. And what the disease we are primarily studying, the group that I'm working with in the School of Dentistry is osteoporosis. And we are studying various mechanisms. I work closely with the biologists. And from the engineering perspective, we are looking at bone remodeling. That's where bone is resorbed and also new bone is formed. And that's why the title is The Role of Mechanical Strain in Bone Forming Pathway Activation in Osteocytes. And clearly this involves both in vivo and in silico. We cannot measure some of these things at the length scales that we have talked about at the nanometer levels inside a mouse model or a human body. So we are replacing that with some in silico simulations to see what is happening. It's a large group of collaborators. We have Dr. Dallas and Dr. Johnson. They are from the School of Dentistry, imaging specialist Deanne, and there are a number of PhD students, animal specialists, Anita, Mark Dallas, and a number of medical students. Gabe is a medical student, and there's also undergraduate collaborators in this project. Just the basics, bone, it's a hard tissue, but believe it or not, there's a lot of cells in there. So there are three types of cells, namely the osteoclasts, the one that eat bone, osteoblasts, wherever the bone's eaten, they go there and form new bone, lay the collagen and also the minerals. And then these osteoblasts get embedded within the bone and eventually get converted to osteocytes. And in the last decade or so, it's believed the osteocytes are believed to be the sensors, mechanosensors. So when you run and when you do all kinds of exercises, they sense the strain in the bone and how they sense the strain and send the signals to the bone cells, osteoclasts and osteoblasts, to eat bone and form new bone. Just like you lay a roadway, you clean up the surface and then put new. It's a very similar pattern. And that's what you see at the bottom is the way they sense it is as you put mechanical load, the osteocytes, they reside in tunnels within your body, within the bone. And these tunnels sense mechanical strain. And there is also fluid around these tunnels between the tunnel itself, cave itself, and the cell. And that fluid is also moving when you're moving. And so they act on the membrane cell surface and then cause fluid flow shear stress. And our role is to see what happens when you apply load, what kind of strains these experience. And from these strains, is there a threshold that will form new bone? So the way we do it is we do mouse model studies. We are studying global strains on the bone by a process called digital image correlation. We've developed some of these techniques in our lab and some of the instrumentation. And you see the instrumentation out here. And this is the biological studies, which is studying the pathway, the Wnt beta-catenin pathway. And you see that there is some cells are blue and others are not. So the question is, why are some cells blue and others not? And that's where we believe the role of fluid flow and shear and strain is. And we do this by a number of different ways. We are developing right now a multispectral confocal image where this is a composite image that shows several things. And what you see here is the 4 different things that from which we make computer models. We can image the nuclei, we can image the osteocytes themselves, and then most importantly, the lacunocanicular network that you connect these osteocytes together. We are able to separate all of them in different models based on this. And this is what you see inside the bone. The blue is the cell itself, then there is a space around it. And you see that it's a fairly complex dendritic network that connects through which these communications happen. So quite often it is, this is one of the more complex models that we have come up with. And from these, we develop in silico modeling where you see the caves themselves. And then as you apply load, we use finite element methods to find the strains within the caves itself. And then we know which one of these is activated because of the beta-gal measurements that we've seen in an earlier slide. And from those depositions of the protein and the strains in these cells, it is for the first time we are trying to connect these two together to directly link a mechanistic biological observation to a physical observation, namely mechanical strain. And so these are the models that we use. And you can see the strain measurements. Some of them are blue, some of them are more, the color coding tells us which is greater in intensity. And then we can compare the strains in each one of these cells, cell locations, and then correlate it, which is what we haven't done yet, but we are in the process of doing it. Correlation, the activation of the pathway to the strains within the lacuna themselves. So some conclusions. This is clearly a work of a multitude of researchers with hundreds of years of experience between themselves and different talents. Otherwise, I cannot be talking in front of you, cannot be standing here and saying anything in front of you. And we use different staining techniques. Some of these techniques we are trying right now and seeing which one works. And with that, I guess I will conclude my presentation. This is currently supported by 2 different projects from the NSF and the National Institute of Health, the NIA Institute. Thank you very much.
The last theme for our research summit today has to do with sensors. Dr. Kim from Missouri S&T is going to talk about biomedical sensor and actuator research.
Okay. Yeah, I'm getting hungry. I think we deserve a good lunch. Yeah, here, the reason I'm here is actually just trying to represent to introduce my excellent colleagues in our center. We have a center, a campus-wide center for biomedical research. It's called the CBR. It's a website is there. It's basically, it's a multidisciplinary, research center. And the director is right over there. I'm not the director, but I'm just kind of trying to introduce this many excellent faculty colleagues whose research is focused on sensors and actuators. We call it like biotransducers, and this is like sort of expertise of our faculty members in the center. And so these are basically our main research thrust here. It's kind of a pretty unique research thrust list is there because we have one of our strengths is a bioactive or biodegradable glass material, and I try to jot down this the best matching like emphasis area, particularly this UM PMI Precision Medical Initiative and emphasis area. but not limited to that, right? So, and then we have lots of inorganic and organic nanomaterials, and we also have a very, unique traumatic brain injury research activities going on. And then these biotransducers, either sensors or actuators, is what I'm trying to represent here. So here's a kind of a short list of our faculty members who are mostly focused on this particular sensors and actuator research. So here's a lot of like mechanical engineering, electrical chemistry, or yeah, there's a couple of other persons there. But again, this is not limited to this the expertise. So first, like Dr. G. Dr. G is basically a nanophotonics person, and she's developing a lot of good meta surface and meta materials for to effectively detect like lots of different chiral structure of the different chemical species. And she's doing, I think, working on like a DoD funded by DoD project to detect a lot of neural biomarkers. So that's, if you are interested in this kind of research, please contact her. And then she's actually in a mechanical engineering doing lots of optics. So it's kind of unique person. And then from electrical engineering, Dr. Ji Huang is basically an optical fiber person. He's doing all kinds of like physical and chemical, detections and sensors based on like optical fiber structures. And particularly like he's an expert in pressures or strain or stress sensing, those kind of thing. And recently he got a huge grant from DoD, Department of Defense, to develop like smart helmet for brain injury. Sorry, not for brain injury, to prevent brain injuries. And this is myself. I'm currently focusing on more like measuring tissue oxygen. So either from those kind of a surface skin patch, which is what we call like a smart dressing sensors for wound healings or vascularization. And also we are working on some implantable biodegradable sensors to measure like tissue oxygen or other metabolites, which can be degradable inside this tissue, so that you don't need to remove it. We don't need a remover surgery, utilizing this bioactive or water-soluble glass. And then Dr. Nam, basically a bioanalytical person. He's been doing a lot of spectroscopy or electrochemical detection for previously more like a biomass or bioenergy research. But these days he's focusing a lot of his effort on this DoD-funded brain injury to detect brain injury biomarkers. So that's his expertise. And then Dr. Nath, basically inorganic chemist, inorganic nanomaterial like a nanorod or nanoparticles, all kinds of different shapes of nanomaterials. She's been working on like previously more like an energy application, but these days she's working on a lot of this therapeutic application with those nanomaterials, and particularly currently she's doing like trying to develop enzymeless glucose sensor, because typically enzymes are it's not very stable, it's biomolecules, protein. But if we can detect glucose with non-enzyme based biosensor, that would give us a really like a holy grail of like long-term stability for glucose detection. And then mechanical engineers basically, and then he's an expert in the thermal or heat exchange type of nanomaterial. Previously, mostly focused on this energy, or like energy storage or battery materials using this kind of nanomaterials. But these days, he's focusing a lot of, he's spending his time on like therapeutic, like this heat exchange mechanism, trying to develop some like actuation device for like a probe or a catheter for thermotherapy or phototherapy for this cancer, malignant tissue. And then last one, I'm sure this is last picture. She's a DNA chemist. She's developing a lot of unique, very novel like DNA origami type of a molecule. So with this novel structured DNA molecule, she's doing a lot of novel like biosensing and nano manufacturing type of thing for sensors and therapeutic application. So I hope these people can get lots of love calls from you, because they can provide you lots of technical solutions. So, okay. Yeah, that's it.
Next is Dr. Lee from Missouri S&T. Title of his presentation is Development of Real-Time 4D Computer Tomography for Medical Applications.
Well, good morning, everybody. I prepared just a couple of slides, not because it is easy, but it is because it is just 5-minute talk. Okay. Well, I'm kind of surprised because I'm the only one working on medical imaging here, so it's kind of strange, but yeah, let's see. Well, what you see on the slide is very sophisticated and the state-of-the-art medical imaging equipment. The CT scanner. I think you are very familiar with how it works, okay? So on the right-hand side, you see the anatomy of the current, most updated CT scanner made by GE, okay? On the left-hand side, that's the motion of the CT scan when it works, okay? So probably first time you see how it moves, okay? And the weight of this CT scanner is like 1 ton. So the left animation shows the rotation of this CT scanner, which is about 3 times per sec. So if you can calculate the centrifugal force of this CT scanner during a patient exam, it will be like a 30 G. So basically, it is very dangerous motion, even though you don't know that. You guys think that CT is kind of dangerous because of the radiation, but no, it is not. In terms of radiation, it is safe, but for the mechanical thing, no, kind of. Okay, so the point is, okay, we use CT scan for many exams, starting from head to the sole of the foot. Well, mostly it is for a kind of stationary motion of a body. But what about heart? Okay. You know, you can hold your breath like for several seconds, that's fine. But can you say, hey, heart, stop? No, you can't do that. But some patient with a problem with irregular motion of a heart, we need to catch that. But even with that very expensive, sophisticated CT motion, the timing resolution is only 3 times per sec. Only we can see 3 frames of 3D object per sec. That's too slow. We cannot see the real motion of the heart. So here is what I'm thinking. Instead of making this gantry rotate, okay, and I said that this gantry is like 1 ton, okay, and the X-ray tube is very big, it's huge, and very heavy. How about making this X-ray tube very small, okay? And if we arrange these X-ray tubes around the gantry, then utilizing this kind of concept, you see the animation over there, then we shoot the X-rays in sequence around this gantry. Then there is no moving part. So that means, we can rotate, we can shoot the X-ray beams more than 3 times. So my first goal is to achieve 30 frames per sec. That cannot be done with the current technology, but if we can develop this kind of a small X-ray tube, okay, then that will be achieved. So, well, the technology is not so easy because we need to shrink this very big X-ray tube to a very smaller one and still need to produce same amount of X-ray photons. So we are doing a lot of simulation study. We are making very good progress. So the issue is how to handle the heat generated during the process of X-ray generation. And we are making some solutions on that. So this idea was recently funded by Army Research Lab through LWI. And we are almost done with the designing stage of this X-ray tube. And we will do a vacuum breakdown test. And hopefully next year at this time, we will see some prototype of the X-ray tube. And maybe 1 more year, then we will make the prototype of this 4D CT prototype. And this will be a very big breakthrough because the size of this CT business is humongous. So thank you.
So the last presenter for the day is Dr. Mark Skubik from Missouri, Columbia. The title of her presentation is Aging in Place Sensor Technologies.
Hello, everyone. Thank you for sticking it out till the end. I'm going to talk about how we're using technology to help our aging population. So what do people want as they age? If you ask them, they will tell you that they want to stay in their own home, or at least they want to have options of where they live. They want to be able to choose where they live. In order to do that, they've got to be healthy enough and functionally independent enough to be able to accomplish that. So what can technology do to help facilitate that and let people really stay in their own homes? So we're going to look at a part of that, but I actually have seen many ideas around the room here about other ideas that can be used to help this too. So when I first started working with our clinical collaborators, especially the nurses, they tell us that people age in a stair-step fashion. If you plot their functional ability over time, they'll go along on a plateau for a while until some dramatic thing happens, like a fall, for instance, and it drops them down to the next level until the next dramatic thing happens. Well, if we can recognize the beginning of that decline, or better yet, predict when that decline is likely to occur, we can go in with an intervention and hopefully keep that functional ability up at a high level. That is, proactively treat or intervene to keep them up at a high level. So we sometimes call that squaring the life curve. We're not necessarily trying to extend somebody's life, but we're trying to extend their health span. We're trying to keep them as healthy and independent as possible, which also helps improve their quality of life and also ends up saving healthcare dollars. So we're trying to turn that red stair-step trajectory into something that looks a little bit more like that green line, up as close as possible. So the way we've been addressing that is by developing a system of sensors that go into people's homes. And we've been doing this for a number of years now, actually since about 15 years, actually. And we've tried different sensors over the years. I'm sure that there's still other sensors that we can add to them. But the ones that we've been using currently include a depth sensor, where you get a distance for each pixel. We use that for being able to detect somebody falling down, as well as to be able to capture movement and walking gait in the home and be able to monitor their walking gait without requiring them to wear any sensors. We have a bed sensor that goes under the mattress. So Dr. Gudaboni talked a little bit about this earlier today. That captures pulse rate, respiration rate, restlessness, general sleep patterns without requiring any on-body contact. And we're working with Dr. Gudaboni to be able to see what else we can do. And we've got motion sensors that we can capture general activity level. We automatically generate alerts and fall alerts and send those to the nurses and the social workers. And now we're exploring how we can send them to consumers like the family members directly. So I'm going to show you a couple of examples. Here's a case of somebody who had pneumonia diagnosed, but 6 weeks before that diagnosis, we got an alert from the system. In this case, it showed bed restlessness. And you can see how that graphical visualization shows it increasing. But of course, these were computational algorithms that automatically recognized that and generated an alert. Here's another case of a change in gait and the alert that we got there about 3 weeks before a fall. And in fact, as we've researched this, we know now that a decrease in walking speed of about 7 centimeters per second over a week means that there's an 86% probability that you're going to fall within the next 3 weeks. So if we can get an intervention in there, we can help prevent those falls. So what we found with our research at Tiger Place in Columbia is that, in fact, if you have these in-home sensors in your apartment with these automated alerts, you can actually have somebody stay there on average 1.7 years longer than if they do not have the alerts. So there's additional research questions to be addressed. What information is clinically relevant? We certainly need lots of other sensors, different features. What level of abstractions do we want to consider? How do we detect very early health changes? And it really comes down to learning a personalized model in a very noisy environment. How do we facilitate early interventions? I think, first of all, we need some additional interventions, but then how do we display the alerts, and who do we display them to? And can we actually suggest an appropriate action? Can we learn enough to be able to suggest what to do next? So there's a number of collaborators from across many disciplines that have contributed to this work. I also want to point out that we have now commercialized this with a spin-out company. This is being run by George Cronus, who's highlighted there. Foresight Healthcare is an MU alum who's now based in St. Louis. So thank you very much for your attention, and I'd love to talk to some of you if you have some ideas.
Wow, I don't know about you, but these were 19 presentations, outstanding presentation of excellent research, and it's impressive that one thinks that we literally have thousands of projects of similar quality going on across our 4 universities.
Just wanted to say a couple of things and then ask questions of you and first of all, so that the audience hear you.
The mic can walk to you.
Yeah, good, thank you. I want to say that this is the 4th, I believe, of the research summits. My goodness, how it's taken off. This is by far the best. Maybe the 5 minutes was a terrific idea. I think I would call them TED Talk teasers because they're so short, but I think it's tremendous what they've grown into. I was disturbed by the last presentation because of that precipitous drop off I think I'm the oldest one in the room. But the question I have to ask is, the slides were terrific. And this is largely driven by the desire of the university to be more collaborative, not only within your department, or your campus, but across the campuses. And it would be a great idea if these PowerPoints could be distributed to those that didn't attend, maybe to the provost, certainly to the various schools or colleges, even the departments. And if those of you who did this here have particular people on other campuses that you think might be interested in collaborating, you can independently send it to them. But this is the kind of inspirational meeting that if we can get that information out, I think you'll get much more interest in collaborating than just us sitting here in Mala. But I think it was really terrific. Thank you, Mark, and Moon, and you. We've not met before, but what a great host. Thank you.
Thank you, Curator Phillips.
I think it was very successful, but I'm biased. But I think what I saw was the possibility for collaborations are amazing. I saw so many talks that even with my limited knowledge, I could see that the possibilities for collaboration existed. Not just across campuses, but even inside the same campus. It is my experience that sometimes as researchers, we're so focused on what we do, we don't know what the person across the hall does. And this was an opportunity to do so. Now, to echo the curator's words, the ones of you who just brought a flash drive, if you can bring it back, we can put your presentations on this computer. I have everybody else's, and then we will put them on a website and give everybody the opportunity to see them, even if you want to work together. Sometimes the contact information flashed really fast, so you will be have an opportunity to go through them again and maybe work on the collaborations. Now, I know I'm standing between you and lunch, and that's not a good place to be. So during lunch, you're going to have an opportunity to sit next to somebody you want to work with and want to talk with and ask questions. So I appreciate you all coming. Thank you for the audience. Thank you for the people who presented. And thank you for our curators who visited and the President Moon who joined us for the last part of it. I hope you saw something that was very exciting. Thank you so much, everybody.